In science fiction, one of the technologies at the top of all of the tech trees are self-replicating nanobots. These tiny machines are too small to see with the naked eye, but can perform almost any function. They can take one form of matter and turn it into another. They can repair themselves, replicate, and they can send and receive signals, and interact with their environment. All of this sounds amazing, but here in reality there are already systems that fit this description. We call them biological life. Biology is pretty much the closest thing we have to alien technology in the sense that we have no idea how it works because we didn't design it, and it's extremely complicated. Even the simplest life forms are capable of amazing feats, not unlike the nanobots of science fiction. Microorganisms are capable of converting almost any carbon and nitrogen source into a nearly infinite array of complex polymers and catalysts that exhibit some truly remarkable properties. And as the organisms get larger and more cells are able to work together, the complexity only increases. Through the use of modern genetic techniques, we've become adept at reprogramming various kinds of life to make new, customized compounds that suit our needs, or produce things in new and exciting ways. From goats making spider silk to glow-in-the-dark beer, the possibilities are truly endless. But if you think back to your biology classes, especially in high school, it probably didn't feel very exciting. Sure, it's obvious that there's enormous levels of complexity, but the impact and capabilities of biology really aren't made clear. You're just left to cram information, regurgitate it later, and maybe water a bean plant. So let's take a different approach and see if we can change that. At its core, doing biology work requires a couple of things. First, you need some kind of organism that you want to grow. This could be anything from plants to animals to that weird mold growing in the bathroom that's gained sentience. Most of the time we grow particular organisms because we want something from them. Farmers grow plants and animals that we can eat, scientists grow bacteria for genetic work, and brewers grow yeast to turn sugar into alcohol. In this video we're going to be focusing on that last one and working with regular Saccharomyces cerevisiae, also known as common brewer's yeast. The reason we're starting with yeast is because to brew wine or mead properly, you need essentially all of the same skills you need to work in a microbiology lab. But unlike a micro lab, a mistake won't accidentally release the plague since our organism isn't dangerous. Best of all, the end product is both edible and delicious if made properly. Next, we'll need a food source for the organism. If you're growing plants or microorganisms, this is usually called your growth media because it's not only their food source, it's also their home. Yeast require a few things to be able to grow. First, they need a carbon source. This is usually in the form of a simple carbohydrate like sucrose, which is a disaccharide, or something more complex like pectin or other starches, which are polysaccharides. Ideally, you also want to provide them with a range of micronutrients and minerals, but for a quick and easy ferment, this isn't strictly necessary. The simplest ferment is sugar and water with yeast mixed in, but it won't taste very good, and the amount of alcohol you'll produce isn't very high. I'll be providing my yeast with both carbon and micronutrients by using whole fruit as my starting material. Here I'm using Chilean pepino fruit, which I can only really describe as tasting tropical? They're part of the nightshade family like tomatoes, but have very bitter skins, so you can only eat the flesh. They're absolutely delicious and ended up making amazing mead. Here I'm chopping them in half and scooping out the flesh, removing the skins and seeds, then adding them to a large pot. I also added some apples and a couple slices of orange for flavor. Cover everything in distilled water and then let it boil for an hour or two, mixing occasionally until everything is a homogeneous mush. What we're doing here is breaking open the cells of the fruit and allowing all the starches and sugars to come out into the water. You can use almost any fruit or source of starch for this, and the taste will heavily depend on what you use and if you do any secondary processing to it. If you're feeling extra lazy, you can skip all of this and just buy a couple of bottles of fruit juice from the grocery store, but it won't taste as good. Now let's talk about the container and what the yeast will do once we start feeding them. The interesting thing about yeast is that they can live in all sorts of different conditions and will produce different things based on what conditions you keep them in. To make alcohol, we need to keep them in an anaerobic or oxygen-free environment. As our yeast eat, they produce CO2 which will build up in the container. We need a way to let the excess CO2 escape while also keeping the oxygen out, so the container doesn't explode. To do this, we use a container that has an airlock. An airlock is basically a one-way valve that uses water to prevent gas from moving in the wrong direction. Some containers come with these built in the rim, like these fermenting crocks. Here we're using a 5-gallon container with a special lid. There's an O-ring in the top where a cheap airlock can fit in place. Before our container can be filled with the growth media we prepared, we need to go over sterile technique. The growth media we made is an ideal food for many microorganisms, not just the yeast. So to make sure that the yeast is the only thing we have growing, we need to make sure everything is extremely clean. The nice part about making wine or mead is that if you make a mistake, the starting materials are fairly cheap and it's obvious when something goes wrong, so starting again isn't a big deal. This is the reason we chose to start with brewing, as it's an ideal way to learn the basics. We start by making our work surface, our hands, and all of our tools sterile, or at least very clean. Wine and mead are a lot less picky than anything you'd work on in a lab, so your sterile technique doesn't need to be perfect, but to hone your skills, it's best to try and be as clean as possible. To start, we spray down our surfaces with an antiseptic called chlorhexidine. This is a 0.02% solution in distilled water. It's very easy to get this stuff online, but 70% or higher rubbing alcohol will also work if you can't find it. For our tools, first we clean them normally with soap and let them dry. For microbiology, this is normally the part where we put everything in an autoclave to make it truly sterile, but for making wine, that's extreme overkill. Instead, we'll be using a special cleanser solution meant for brewing. Our chlorhexidine solution from before will also work, or spray everything down with rubbing alcohol. Make sure that no matter what you use, everything is fully dry before using it with our yeast. Finally, before we handle anything, we wash our hands, then put on a pair of gloves, spray them with chlorhexidine or rubbing alcohol, and let them dry. Now we can start adding our growth media to our jars, but first we need to filter out the large bits of fruit that are left over. To do this, we cover the mouth of our jars with some clean cotton cloth and secure it down with an elastic, leaving a divot in the middle to make a simple funnel. Slowly pour the mix we made earlier into the funnel to separate the solids. As the solids collect, the filter is going to get very full, so you're going to need to stop occasionally, lift out the funnel, and squeeze out any remaining liquid, before scraping our solids into a collection bucket. Don't throw these out, as they're still useful for cooking. We turned ours into a spicy apple pepino crumble when this was done. Keep filtering until all the liquid has been processed, and save a little bit of the filtered liquid in a separate clean bowl on the side. Any cloudiness that remains in the solution at this point are the starches and pectins, and will get broken down as the yeast eat. To the liquid we put off to the side, add in about a teaspoon of active dry yeast. This is going to be our starter culture. Give it a quick stir to disperse the yeast, and then wait a few minutes. The yeast will wake up and start eating the sugars and produce little bubbles. While we're waiting, we're going to increase the amount of sugar in our solution. The more sugar we add, the more alcohol will be produced, up to a point. Here's where we get to the fundamental difference between wine and mead. For wine, you'd add sugar, but for mead, you'd add honey. Just like our choice of fruit, our choice of sugar source will also change the taste. For our 5-gallon jug, we add one large container of honey, about a half liter in total. Honey is sterile, so nothing needs to be done to it before adding it to our mixture. Make sure everything is well mixed, then add the starter culture we prepared earlier. And with that, most of the work is done. We place the container somewhere warm and out of direct sunlight and let it sit for anywhere from a week to a month. The longer you leave it, the more alcohol will be produced and the less sweet the final drink will be. As it begins to ferment, you'll see bubbles coming through the airlock. After several weeks, when the bubbling stops, it's probably done. After about 5 weeks, we tasted the brew and decided it was finished. You'll notice that there's a large amount of sediment at the bottom of the container. This is all the dead yeast that accumulates over time, but it'll taste bad, so we're going to need to remove it. To do this, we carefully pour off the solution on top and leave as much of the yeast behind as possible. This is called decanting. I wanted to be fancy, so I decided to mull my mead with spices. In this case, ginger, cayenne pepper, cinnamon cloves, and nutmeg. This is as simple as adding the spices to the solution that we just poured off, putting the lid back on, and leaving it in a warm place for another 2 days. After that, it's quickly filtered again and bottled. If you want carbonation, add in a small amount of sugar before bottling. This will give any remaining yeast a bit of food so that they can produce more CO2. Since there's no airlock on the bottle, the CO2 is forced to dissolve in the solution, carbonating it. When the pressure is released, all of the CO2 can come back out of the solution, giving you a bubbly drink. Be careful not to add too much sugar or the gas buildup can cause the bottle to explode. And with that, we've not only produced a delicious adult beverage, but we've also covered almost all of the basic biology skills. From media preparation to sterile technique to organism care and control of the growth environment. With the basics down, you're now one step closer to being a biologist. From here, doing other ferments is easy, and I'll show some other simple recipes in the future. Depending on which organisms the ferments use, we'll need to provide different conditions and food sources for them. Next time, we'll also get a little more advanced and look at how to make media for bacteria, and how to pour and streak plates. After that, we can start looking at growing organisms for research and genetic applications, as well as even more advanced growing techniques like using a bioreactor. I'll be releasing a new video every other Monday, so be sure to check back for all of that. Most of the footage for this video came from my time studying with Gabriel Lucena. He's an accomplished microbiologist, and I learn a lot from this whole process. Recently, we've joined together to form SciHouse, a makerspace in Jacksonville, Florida, where we'll be able to continue teaching and finish much of the work we started at this course. So be sure to keep an eye out for all of the new content we'll be making there. I've added some links to SciHouse's various pages in the description, as well as a link to the first SciHouse video. Also, if you enjoy biohacking and transhumanism, be sure to check out the guys over at the Dangerous Minds podcast. I got a chance to sit down with them a while ago, and we ended up having a great conversation. They've had all sorts of interesting people on, so I'd highly recommend checking them out. That's all for this video. I hope you've enjoyed. If you have, be sure to subscribe and consider clicking the bell to get email alerts when I post new videos. If you've got ideas for other biology projects we should try, be sure to leave them in the comments down below. I love reading your suggestions. If you enjoyed and would like to help support the continued production of science videos like this one, I'd really appreciate if you check out my Patreon. That's all for now, and I'll see you next time.